talk. So we move on for the second talk today, and it will be given by the Professor Alessandro Varani from Unesp, and he will talk about exploring the bacterial transposases diversity from metagenomics data with ISsaga. Yeah? 2.0? Okay, Alessandro, your time.
Bom dia a todos. Gostaria de agradecer o convite. É uma oportunidade muito boa para poder apresentar para vocês um pouco do trabalho que eu venho desenvolvendo nos últimos anos. Now I'm going to switch to English. And first of all, I would like to say that this is an ongoing project. It's not a finished yet project. This is about one pipeline which were developed to study transposases in metagenomes, okay? And let's go and start to talk about transposases and bacterial genomes and metagenomes. This is the outline of the presentation. And the first stage I would like to show up a little bit about the story about the transposase, where you can find it, and the importance and the diversity of the transposases in bacterial genomes. I am in the prokaryotic world. And I will talk about a little bit why to study transposases in metagenomes and why it's important. Show up some examples. And in the end, I will show the pipeline, ISsaga, which I developed in the last year, which were first developed to study in complete genomes, and now I'm going to try to develop a new version for metagenomes, showing some examples and a little bit of the beta version, which is not released yet, but it's more or less functional. Well, talking about transposases, and we need to start to think about where you can find transposases in prokaryotic world, and the major sources are the insertion sequence. Insertion sequence are the shortest transposable elements found in bacterial genomes. They are components of the mobilome. And when we talk about mobilome, we are talking about a lot of different elements which can be found in bacterial genomes. Could be genome islands, prophages, conjugative transposons, superintegrons, unit transpose, compound transposes, and the insertion sequence here. As you see here, in this figure, we have one scale of size, and indeed the insertion sequence are the shortest one. There's a little joke about the stable genome, the stay-at-home genome. This term was stated by Peter Young in 2003. And the mobilome, the term was stated by Ariane Toussaint in 2003. And when we talk about mobilome, they represent in some genomes a large amount of these genomes. For example, in Ecology and Shigella, they have a common 4 megabases, but the islands and the mobilome are the source of the major variation between these genomes. And we can see it in a lot of different genomes, this big variation caused by these mobile genetic elements. And in this talk, I'm going to talk about the insertion sequence and how to analyze these things in genomes and how we intend to analyze it in metagenomes with this new tool, ISsaga. Well, it's not so simple. Insertion sequence, if you pick up the genetic books, we have one example on the test books showing the insertion sequence. They are formed by one transposase gene, inverted repeats on the borders, as you can see here on the figure, and the target site duplications caused by the insertion of these elements. Okay. Regarding the number of genes here, could be 1 or 2 open reading frames. The transposase and some regulatory protein can occur. And the inverted repeats are most of the time imperfect This means that it's not exactly the same composition of these inverter repeats in each border. And most of these elements have a small flank tangent duplications. Okay? This is the classical textbook, but it's not so simple. We have a lot of different examples, a growing number of atypical insertion elements. For example, we have insertion sequence which transposing using single-strand DNA. And here is one of these examples. And you can see here are palindromic regions. Okay, it's not inverted repeats. It forms a secondary structure. And with different transposases. We have also another different insertion sequence with no inverted repeats and no target side duplications, direct repeats. And we have also some other difference with accessory genes, for example, in this family. And I'm going to show in the next slides the number of different families which we already classified. And we have also different classes of insertion sequence with passenger genes. These guys here are really interesting because they are between the frontier between insertion sequence and transposons. Okay, we have this kind of difference. In fact, we can go to something like that. This is the classical element. Each part here represents the invert repeat, the transposase. And what we can find in bacterial genomes is in some events of deletion or pseudo gene formation, we can have the mites or pates. Pates means palindromic associated transposable element, and mite miniature inverted transposable element. This can occur a lot of genomes, and most of the time this is forgotten in the annotation programs. We can also have passenger genes forming which we call TAS, the transposase, and two passenger genes in this example belonging to these families. And we also have one very interesting thing. Some of these elements can lose the transposase, and we have one thing called MAKE, which is very common in bacterial genomes, and which is really, really complicated to annotate these guys, because we need to find out the mother element with the inverted repeats to find these guys in bacterial genomes. Very interesting. We have it, for example, in Xanthomonas. A lot of pathogenicity genes are flanked by inverted repeats for one transposal from TN3 family. This is very interesting. This is one part of our projects here, studying these elements. And all of this, we try to classify all these elements regarding the composition, invert repeats, direct repeats. We define the families by this organization, by the alignment of the transposase, by the sequence of the ends of the borders, the beginning, the end of the insertion sequence, the presence or absence of direct target repeats, but also, which is very important, we can classify these elements based on the enzymatic activity of the transposase. This means how the transposase cleaves the DNA during the transposition process. And there's a lot of groups studying this mechanism. Well, talking about the transposase, we have three major types of transposases. I don't know if I can say that, but most of the transposases found in bacterial genomes are the DDE transposase. Almost all transposable elements have this kind of transposase. The most important fact is that they do not make covalent enzyme substrate intermediates, okay, and use water to break the DNA. We have the HUH transposase related to rolling sarcoplasmids and viral wrap proteins. And in this case here, they make a covalent enzyme substrate intermediates. And generally, they use a tyrosine to break the DNA during the process. And we also have site-specific recombinase, which can be one tyrosine or serine, residue related with the catalysis of the movement. And this make covalent enzyme substrate intermediates. That's the most important transposase. And using that criteria that I showed before, we can classify these elements in 28 families already, and one classified group, which is this one here, IS non-classified yet. The different colors here means the different subfamilies. Okay, this means that this family here we have 1, 2, 3, 4, 5, 6, 7 subfamilies based on the alignment of the transposase. We have these families, and in the last year we have a growing number of these families marked here on the test, which is very important when we talk about metagenomics. We are talking about a lot of data, which can certainly can be used to help us to classify it and, well, maybe find another new families or help us to classify this. That's why we are looking for transposases in metagenomics now, one of the reasons. And when you look at the distribution of this insertion sequence in the prokaryotic world, we have something like that. Here is the families, bacteria, archaea, and we have, as you can see here, most of all the families found in all groups of bacteria. And just a little on the archaeas. Well, what I'm going to show here is one database dedicated to catalog all this insertion sequence. This database is called ISFinder. It's based on France, in Toulouse. Okay, this database is maintained by my collaborators. Okay, I'm a collaborator of the ISFinder database. This is the face of this, the front page of the database, ISFinder. This is the old web page. We have now a new database, a new web page with a lot of new features. What I'm going to show in the next slides. In this database, we have information about each family, information about, you can see here, DD motifs, the different families. We have the catalog of our insertion sequence already found in bacterial genomes. We can also do search looking for one insertion sequence in particular for one genome. You can see here we can choose, for example, the genera, species, family, for example. We have also one blast search. And the most important thing, one of the most important things, we have one submitter form, which means that the user can also submit one new identified IS to ISFinder database. During the last 5 years, we also developed a genomic section, which is pretty nice interface, where we maintain one curated database of annotated insertion sequence in complete sequenced genomes. And if you can choose one letter here, I can open one list of replicons, of genomes. And if you can choose one, for example, sulfolobos sulfataricus, it's very teeny letter here, but this is a sulfolobos sulfataricus. And we can see the circular map, genomic circular map, showing all the insertion sequence found in one genome. Here in blue, we have complete elements, and in gray, light gray, we have the partial elements, which are also very important and most of the time forgotten in the annotation programs, the partial elements. And as you can see here, we have a lot of partial elements. When we look about the number of insertion sequences already annotated, identified by ISFinder, we have this chart here, this growth here. In 98, we had only 50 This was released with the first book, Mobile DNA, and after some publications, the beginning of S-Finder, and most important, the beginning of genome analysis, when the S-Finder staff start working genomes, annotating genomes, this number is become much more higher. This number is for 2010. Nowadays, we have more than 4,600 different ISs already identified in bacterial genomes. But when we estimated the total number, looking at the HalfSec database in GenBank, we estimated 35,000 different ISs. This means that there is a lot of work yet for the people which work in annotating these elements. We have a lot of different ISs yet to be annotated, only looking the complete sequence genomes. This is the new website, which I said before, which will be released next year, completely rewritten to permit additional other types of transposable elements. Just a quick look of the new website, which will be released, as I said, next year. And now the question, why I came here and show up to you guys the transposases, and why it's important to study transposases in genomics and mainly in metagenomes. Well, I'm going to show four reasons which are interconnected. The first one is the role of these elements. They shape genomes. Large chromosomes rearrangements, insertion mutations, gene activation and inactivations, and they are natural vectors for horizontal gene transfers. Only look at this point, we can think about and we can figure out that, sometimes the gene which we are looking in our metagenomes could be a product of lateral gene transfer, or one other example, or one transposable could be inserted in the promoter region before your gene, and this action can change the expression. Well, this is important. But also, the ubiquity, because the transposases are by far one of the most abundant genes found in nature. And this suggests that these transposases, these genes, can play a preponderant role in life. And this suggests also that what we know about the transposases, looking only at the sequence of the genomes, could be just a tip of one iceberg. We can know just maybe just a little about these enzymes, and maybe can exist much more transposases, and we don't know yet. And I really like this article here, because the authors looked for the abundance of the genes, the functional roles of the genes in genomes and metagenomes. Look in genomes, we see the transposase just over here, in a very high position compared with other genes, transcription regulators, ABC transporters, etc. And when you look for metagenomes, and this figure where I showed it yesterday, we have the same thing. The transposases are really abundant. They are really, it's really easy to find the transposases in metagenomic projects. And this means that if they are abundant, we can maybe assemble them and try to reconstruct the complete transposase. Third reason, diversity. And this is really amazing, because the more we learn, the less clear are the distinctions between different transposable elements. This means that the classification of these elements, as I showed before, the difference, the mites, the makes, we always are found something different looking at the genomes. Each new genome, sequence genome, show up to us different thing about these elements. And thinking about metagenomes and the amount of data which can be generated, this means that I'm pretty sure that in the future we will find completely new variations of these elements. They are very diverse. And when we look at the transposase domains, we find something like that. The transposase like Lego modulus. Here we see different families and the domains of these families. And even inside the same families, we have variations like that. We have transposases with one open ring frame and transposase generated by a frame shift frame shift. And the domains, as you can see here, in each of these families are variable. And the idea now, which we have, that these enzymes are formed by different modules. For the reason is that they are underestimated. This means that most of what we know on the diversity of these elements and their are based of a specific interest of the research lines, such as antibiotic resistance or capacity to metabolize specific compounds. And despite the huge amount of data and the genomes and metagenomes available on the public databases, the real impact of these elements and their role on the microbial world most likely remain underestimated. We really don't know much things about the impact of these elements, even in complete genomes. And you cannot imagine what the impact of these guys in metagenomes, looking metagenomics data, and the opportunity to look at this large amount of data generated by the metagenomes project. Well, thinking about the impact of this insertion sequence in bacterial cell, the central role in assembly, sequestration, transmission of a group of accessory genes. Well, they can bilateral gene transfer, change genes between different strains. Resistance to antibiotics, capacity to use on unusual compounds as are carbon source. And they are also associated with symbiosis, pathogenicity, and virulence. They have a general role in the genome remodeling, causing deletions, inversions, and replicon fusions. But in general, these elements improve the adaptability of the bacteria to respond to changes in the environment. Well, just looking at these points here and think about and to analyze metagenomes is something which really need to look at. Why not? We already know a lot of things about these elements, these transposable elements, so why not to look at that on metagenomes projects? Let's see some examples in complete genomes. Before this one, I have shown it before. What is proposal about the expansion of this element and niche restriction? Let's think about one ancestor genome, and here is one insertion sequence, one transposable element. And after some modifications in the environment, like genetic isolation or the changement of the environment, could occur expansion of these elements, okay? And this expansion generally, what happens is one transposable elements inserting side genes, and this during the evolutionary time scale can generate pseudogenes and deletion of genes, and therefore causing a genome reduction. This genome reduction among the time will eliminate also the insertion sequence, and we have a streamlined genome without these elements, which occasionally can occur a reinfection of elements and the cycle repeats. And this process increases the host dependence. And this is what happened in some example of pathogens, which are now restricted to one specific host. Let's see some examples of the impact of these elements in complete genomes now. For example, Shigella is a completely insertion sequence jungle. We see here different strains of Shigella, and looking at some different insertion sequence in Shigella. And as you can see here, there is a completely different patterns of these elements in different strains of Shigella. This means that if you pick up the Shigella genomes now and try to analyze these elements, you see a complete different pattern for each Shigella on the database. Other example is the virulence plasmid of Shigella, which this plasmid here, the green dots here represent insertion sequence. And when you look at the virulence of Shigella, the virulence genes are flanked by insertion sequence. For example, here, there's some virulence genes, and here, up here, some virulence genes. And when you look when you analyze by simple blast these elements here, the first hit are related with E. coli and other enterobacteria, which means that maybe these plasmids were assembled and passed through different enterobacteria before to be part of Shigella flexerni. Other example, one very nice example is Bordetella pertussis, and here is the comparison of three different species of Bordetella. Okay, each triangle here represents one insertion sequence, and as you can see here, this strain have a lot of different insertion sequence compared with this other here. And what happened here is a genome reduction in pertussis genome. And together with the genome reductions occurred also a restriction of the host. These strain here can cause disease in different hosts, but these strain here are restricted for only one host, and this were caused by the insertion of this element and by the expansion of these elements. And one last example is Yersinia pestis, genomic inversions. When you plot the GC skew of the genomes, most of the times the GC skews help us to find out the beginning of the replication of one genome, of the terms of the replication. But the GC skew can also show up to us lateral horizontal transfer events, or at least can show hot points for lateral transfer events. And when you look at the GC skew of Yersinia pestis, we can see that there were some inversions of GC skew. And when you look at the borders of these inversions, we can find different transposons elements. Okay. This just some examples shown up the impact of these elements. There's a lot of course, there's a lot of other examples, but just these three examples. And the next question maybe you guys can ask is how to analyze the mobilome, how to analyze these elements in bacterial genomes. And one big problem is that mobile genetic elements annotation escapes all the traditional genome annotation methods in general. We are talking about different ways to annotate and identify these elements. In general, we use one gene predictor, like Glimmer, Prodigo, which are very nice to find genes, but about mobile elements, it's just the beginning. What do we have now about identification and insertion sequencing genomes? We have one program developed by Wagner Group in 2007, ISsaga. Which is written in Perl scripts and depends on a previous created IS as input. And this is a limitation because we need to identify the IS to use the software. Okay. We have Oasis, which is written in Python and does a de novo prediction, employing ISFinder database for transposase function. This is a pretty nice software. And we have ISsaga, which I'm going to talk now in the end of the presentation, which is integrated in ISFinder database. It's a web service made in Perl and PHP for web interface. Uses a de novo prediction, employing ISFinder database, and also provided tools for manual annotation and creation. As I said, this software only help the beginning of the annotation of these elements. Some manual assessment is essential, always, to identify the ends, the invert repeats, the target side duplications. And this is really important to identify these elements, because with this identification, we can find also how occurred the transposition and how is the nature of the biochemistry of the transposase dependent of the presence or absence of each of these elements. Well, none of these softwares were developed for metagenomic data, and the most important point here, current metagenome platforms does not deal with transposases in fine-scale classification. Most because we are talking about short reads generated by Lumina technology, IOTorrent technology. Very small reads are not enough to identify the complete family and domains. That's why important to assemble these metagenomes to try out to identify them. Talking about ISsaga, the first release was in 2011. This is the address of the platform. And some numbers about these platforms. We have already 25 citations, and this number of users from different parts of the world. We have more than 1,000 annotation projects already loaded on the platform, and more than 4,000 different genomes are already analyzed by this platform. It's currently used by a lot of people. And the idea now is to integrate the metagenomic feature on ISsaga, is to provide a way to, with your metagenomic data, if you want to figure out how many transposases, is there a way to analyze the lateral gene transfer on my metagenomic data? The idea is now, in this new version, which I said in the beginning is a work in progress, is not already done, to use this platform to analyze in this point of view your data. And how it works, ISsaga? We can submit your genome file or metagenome file. We have here in this color automatic process, and in this color in blue manual process. You submit your genome file. There is a transposase detection, which you generate annotation tables. Okay. Now, after this part, after the protein part, the detection of the transposase, we have a second part of the pipeline in manual process. And in this manual process, the next step is to look in AskFinder database what we already have annotated. The question is there any insertion sequence already on the database which I can use to annotate in my new genome? And this part will do that. We'll generate annotation reports, which will update the annotation tables. And on the other side, we have annotation of new elements. ISsaga have one predictor, which generates FASTA files, with potential insertion sequence, new insertion sequence. And use this part of the pipeline, you can annotate it in new IS, which will update the annotation table. As you can see here, it's not automatically processed. Is what I said in the beginning. It's really difficult to generate and write one pipeline to do everything automatically, even because we don't know exactly all the diversity, the 4 reasons which I showed before about these elements. And this will generate the final annotation. One example of one run in one genome. This is Cosmotog olearia, one bacteria. I loaded this genome on the platform, and the first result is this pie chart showing the families. Very interesting here because we have one family not classified yet with one considerable number of transposases of this family. And the second feature is the predictor, which is really nice, this part, because the system can show the number of open reading frames related with transposases, the number of complete transposases, the number of partial transposases, self-genes, and transposases not yet classified using AskFinder. And we also have one predictor of the number of insertion sequence, total number, and different number. You want table showing the families and this distribution. This will be implemented in the metagenomic section of ISsaga in the future. What we also have is one table with insertion sequence already identified in this genome. In this example, I used one genome which already completely annotated, and that's why we have this big list with insertion sequence, which is already, I can use to annotate my genome. We have also one predictor showing circular maps, the position of each of one of these insertion sequence. And in the end of the process, we can generate circular maps showing the real distribution. And if you guys remember the Bordetella Petrucis, which I showed up before in some slides, this is the organization of each of insertion sequences on Bordetella. Some examples, and one good thing in the end, the system can generate one new GenBank file with the comment insertion sequence annotated by ISsaga. This will work for metagenomics also in the future, which can be exploited in other tools for downstream analysis. Talking about the metagenomics support. Release date will be next year, some date next year. I don't have exactly the date. And there is a lot of tests which is necessary to be done before. Requirements. Assembled metagenome in multi-fast format. As I said, the transposases are really ubiquitous in all sequencing genomes and metagenomes. This means that we can assemble these metagenomes and find transposases. One requirement, only contigs with this size will be processed, and there is no number of contigs. And regarding the FASTA file format requirements, will be the same format which we use for ImageRast. I just copied the part of the ImageRast website, but it's the same format. This means that you can submit your assembled metagenomic to ImageRast, and later, if you want, you can submit to ISsaga to find out your transposase. Okay, the main features of the metagenomic support is the Prodigo for ORF annotation in metagenomic mod. The Prodigo, the last version of Prodigo, have one flag which you can choose for metagenomes. One BLAST-X deep scan. This means that after this prediction, the BLAST-X will scan the sequence from the beginning to the end to try to find partial elements. And we have also one ratio between the contig coverage, I mean the assembly, and the assembly metagenomic coverage, which can help us to estimate it, overrepresented transposase. Okay? And we have also taxonomic labels for each contig. These taxonomic labels generated by Kraken software, okay? The Kraken software, which were released this year. And yesterday we have one talk showing that there is maybe other options for the use for this taxonomy prediction, other softwares. Maybe in the future I can use different ones. This is the first look of ISsaga platform. It's necessary one username, is necessary one password. This means that you need an account. If you click here, down here, we'll open one form, and you can fill this form and put your information, email, password, and after one approval, you'll be able to use the platform. Okay, this is the beta version. It's not released yet. Okay. And thinking about the metagenomes, let's take a quick look in this beta version in some metagenomic examples. I'm going to show sugarcane in natural forest cultivated soils generated by our group in Jaboticabal. Okay, we have 4 different samples generated by Illumina HiScan. 45 million reads per end for these 4 samples. Natural forest during the dry season and forest during the wet season, sugarcane dry season, and sugarcane wet season. Okay, these metagenomes were assembled using CLC Genomics Workbench, the last version, genome assembly. And we got this number of contigs which were submitted to ISsaga. Okay, it's a large number of contigs with any 50 of this size. And this is the length of assembled metagenomes of each sample. I loaded each of these contigs on ISsaga, and I got this. This is the first prediction. As you can see here, we have a distribution of different families. This family here is one of the most prevalent. The non-classified yet, just a little. And we have one table just below the chart, the pie chart, showing each contig, size, the coverage of this contig. We also have here the mean coverage of the metagenome. This means that I can compare this number of these, and this is really interesting. As you can see here, we have a very big coverage compared with the mean of the metagenome. This can at least help us to estimate that this contig maybe is overrepresented on the assembly. We have here the number of transposases. We have also one error correction tool to try to show up the false positives. And here we have transposases in the first, excluding potential false positives, and this included potential false positives. This table is very large. There is an option here which I can roll and see all the contigs. And here in the last column, I have the taxonomy prediction using cracking. This is really helpful because during the annotation, I can figure out from which organism my insertion comes from, my insertion sequence. And here, the prediction about number of orths and number of different insertion sequence. We have also one genome browser for each contig. This means that I can look where is the position of my insertion sequence in each contig. We have one first track here showing the annotation and a second track here showing the position of the insertion sequence, and here the open reading frame of the insertion sequence. If you put up the mouse here, we will open a dialog showing the annotation information, okay? And you can zoom up this map and you can analyze where is located each of your transposases. Looking out the forest wet season, we have this distribution. As you can see, we have some difference between these two forest samples, different families. The same things, you can see the size of each contig, coverage, transposase, and taxonomical classification. The genome browser here is very nice because we can see the genomic context of this. Then another sample, sugarcane dry season. The same thing, other distribution, the same features, predictors. As you can see in this, we have around 100 to 200 different insertion sequences in each of these metagenomes. And at last, the sugarcane wet season. This is I really like it because, as you can see here, we have a large number of non-classified yet insertion sequence. And that is what I'm looking for, new transposases to annotate and to make ISFinder database better for the next predictions, which will be made by the software. Predictions, coverage, same things. Okay, this is just the beginning of this new tool, which the idea is to annotate and try to understand the lateral gene transfer phenomena in bacteria and now in metagenomes. It's a promise to be the first tool for yes annotation in metagenomes. And as you can see, there's a high diversity of IS transposases in the tested samples. But there is also several improvements and new features which we are planning to do during the next months before the final release. One of these tools is horizontal gene transfer analyzer, trying to find out compound transposons, transporter ISs, mix elements for target genes, and scars of insertions caused by mites, baits in promoter regions. This means that, okay, you can load your genome image host, and your next question is, is my target gene is a product of lateral gene transfer, or there's something happen in my target gene, in my metagenome, you can use this tool, load your metagenome in this tool, and ask it for this tool to see if there is any of these things occurring in your target gene of your metagenome. That's the idea in the beginning. The release date, as I said, is next year. And I would like to thank you very much, my collaborators from CNRS in France, Professor Eliana Lemos from Cabal, which gave to me the metagenomic data to test this tool, and my collaborator in NIH, which is my beta tester. She's annotating 200 genomes, and during the annotation she's found some bugs and helped me to make this software better. And thank you all of us your attention, and if you have questions, it will be a pleasure to answer. Thank you.
Thank you, Alessandro. I'm open for 2 or 3 questions. One.
Thank you very much. That looks really useful. So, the genes that they carry around, so the potential horizontally transferred functions.
Sorry, can you say that again?
The potentially horizontally transferred functions, or like maybe insertion element cargo genes. Have you looked at all of their functions? Can you annotate most of their functions or not?
You mean about the cargo genes insertion sequence?
Mm-hmm.
Yeah. Antibiotic resistance genes, most of them. But I think that we have a bias there because there's the antibiotic resistance people which work looking antibiotic resistant genes, which found these elements in the beginning, 20, 30 years ago. But there's others, metal resistance and, well, depends of the organism, the environment. We can find different genes located inside these elements. In Xanthomonas, for example, we have a secretion type system, type 3 secretion systems located in these elements. Okay.
And is, so are most of them annotatable, or are there also a whole lot of unknown genes?
I think that most are unknown. Yeah. Only if you have one question. For example, if you work with a type 3 secretion system and you ask me, let's try to look my genome if I can find it. And if you look, annotate it, maybe you can find. The point is, depend of your question, depend if you want to go deep on the annotation. And what I said before, most of the problem is that these elements are more or less forgotten because they escape the traditional annotation methods, right?
You showed that groups of bacteria have groups of IS elements. Have you looked at different metagenomes to see if metagenomes from similar environments share similar IS elements?
Yeah, good question. Not yet. Not yet. This is a future thing which we intend to do. Yeah. Maybe. We don't know.
Hi, I am Felipe from ISsaga. I'm excited about the new ISsaga update. I think it's gonna work. And I would like to ask you, what can we discover comparing the results of ISsaga among different samples? I mean, we have different graphics showing the families and evidence of families, IS families, but how can we say, oh, so this area have more ES than other, and this family is more abundant, but what it means, evolutionary or something?
I didn't get your question. Can you ask in Portuguese? It would be much more easier.
Claro. Queria saber, comparando áreas com o resultado do ISsaga, o que a gente pode inferir, por exemplo, se eu tenho uma área A e uma área B, eu comparo os IS dessas duas áreas. quanto às famílias presentes e a abundância delas. O que eu posso inferir, por exemplo, nessa área, quer dizer que então os genomas estão maiores, menores, está sob estresse ambiental, ou tem como... Porque os organismos têm quantidades muito distintas de IS entre si. Então, dependendo dos organismos que eu vou ter lá e da abundância deles, eu já devo ter uma mudança nos padrões dos IS, não?
Entendi. Bom, o que você pode fazer, e nesse módulo que eu vou desenvolver mais para frente, é estudar impacto em regiões promotoras. Se o elemento entrou numa região promotora que pode ter quebrado um promotor, por exemplo, o que vai impactar na expressão dos genes que estão logo depois dele. Outra coisa é se um gene de interesse está dentro de um elemento em particular. Era isso mesmo a pergunta, né, que você tinha feito. Está dentro de se um gene em particular de seu interesse está dentro de um transposon. Certo? E essa ferramenta vai dar uma lista no final, quando você carregar seu metagenoma. Ele vai falar, olha esse contigo aqui, porque está acontecendo isso, esse, porque está acontecendo aquilo, e assim por diante. Tá? Era isso mesmo?
Era isso. Sim. Então, a ideia é que ele apresente, dê um panorama geral de onde eu devo olhar com mais cuidado e procurar impactos mais específicos. É isso, né?
Yeah, the point is, in this new version, I'll develop one table showing hotspots to look. Because if you're talking about one assembled metagenomes, we are talking about 1,000 of contigs. And certainly, this is really not easy to open each of one and look, what happened in this one, in the next one, etc. But there will be a table showing hot points. Take a look on contig 2011, for example. In this contig, maybe we have one compound transposon with one gene related with, well, some function. Right?
Great. Some other question? No? If not, then you give a break for the cough time, and then you come back at 15 to 11, okay?